Good morning, everyone. My name is Dr. Jennifer Estall. I am a professor at the University of Montreal and McGill in Montreal and work at the Clinical Research Institute of Montreal. I'm really excited to be chairing this morning session which is an annual symposium organized by the Diabetes Journal. And this year it is going to be focusing on the nervous system, central nervous system control of systemic metabolism. It's all in your head. So I want to introduce our first speaker today, Dr. Zaman Mirzadeh. He trained at UCSF, where he completed his PhD in 2008 with a focus in neurodevelopment, and his MD in 2010. He completed neurosurgery residency at the Barrow Neurological Institute and his postdoctoral research fellowship at UW, where he developed his interest in neurophysiology of metabolic homeostasis. In 2017, he joined the neuroscience faculty at Barrow Neurological Institute, specializing in functional neurosurgery. He uses implantable neuromodulation devices targeting abnormal neurocircuits to treat neurological disorders and direct a laboratory-based research program investigating neural circuits involved in hypothalamic control of energy and glucose homeostasis. If we'd like to welcome Dr. Zaman Dr. Mirzadeh to the stage. And while he's setting up, I just want to remind everybody to silence their cell phones. We're going to have a few questions after each speaker. And at the end, if we have time, we'll have a panel discussion. And also, please respect the discretion of the speakers if they want to have photography or not. It will be indicated on their slides, or they'll let you know ahead of time whether it's allowed. Thanks.
Yeah, I think for my slides, you can take photographs. I don't think I'm talking about anything that's unpublished here. My goal for my part of this session is really just to provide a general framework for how we think about nervous system control of glycemia in both normal physiology and then in the setting of diabetes. So, then
There's a thing here.
I may not be able to use it. So, I think everybody here is familiar with this traditional islet-based model of how we think about glucose homeostasis with the various peripheral organs that are involved. And although it's a gross oversimplification, the basic premise is that elevated glucose levels result in insulin release from beta cells, which are glucose sensing, and that has actions on these various organs to restore euglycemia. Now, one line of investigation is, of course, to go much deeper into this and everything that's not shown here, and how does the islet regulate that secretion, what other factors are involved intrinsically within these organs and between these organs that are the basis of this type of control. But for today, primarily what we're going to be talking about is looking at this whole kernel as something that the nervous system can impinge upon. So you have the nervous system that's embedded within all of these organs and can sense both the state of these organs and provide modulation to these organs. And this slide is going to get intentionally a little bit complicated, but just to show how far we've come in this understanding. So we have autonomic innervation that provides output to these various organs and can control both insulin-dependent and insulin-independent mechanisms of glucose disposal. Then there's sensory innervation that's mediated both by spinal afferents as well as by vagal afferents to get information back to the brain. And then, of course, we have central processing in the brain to make decisions about what needs to happen based on these various metabolic states. So now in this talk, what I'm going to do is go through these various components and just highlight examples of why these different components matter in terms of both normal physiology and in the setting of diabetes. And that'll come from both there'll be a mixture of animal and human studies. And we're going to go kind of one at a time. I'm going to first talk about the outputs, primarily autonomic nervous system outputs, and then I'll talk about the central processing, and then we'll talk about the sensory innervation. But before I do that, I'm going to try to highlight why is CNS-based control important. And so this is a challenge that's faced by animals all over the world every day. And that's the environmental challenge of cold exposure. And it's something that's been studied well in the laboratory. So, and it's helped us understand why the nervous system is so critical. So upon cold exposure, there's various organ systems that are involved, and I'm just highlighting a few of them. So we have pancreas and islets, as I'll show you, brown adipose tissue, skeletal muscle, and the nervous system. The first thing that happens is that cutaneous thermosensory receptors sense the changing temperature and relay that information ultimately to pre-autonomic circuits in the hypothalamus. There's a descending flow of information that gets generated and passes through sympathetic nerves and also motor nerves, and that induces BAT thermogenesis, so brown adipose tissue thermogenesis, and skeletal muscle shivering. And this massive increase in energy utilization is powered by an increased uptake of glucose, which is made available by increased insulin sensitization of these tissues in the setting of cold exposure. Now, if nothing else were to happen, then glucose levels would rapidly plummet in this setting because you have a massive increase of glucose utilization and you haven't done anything to account for that otherwise, and you would become hypoglycemic and die. But that doesn't happen. Instead, what happens is that a parallel outflow simultaneously provides sympathetic tone to islets, the beta cells, and that reduces glucose-stimulated insulin secretion, so that insulin secretion goes down. And that coupling of the increased insulin sensitivity to the decreased insulin secretion keeps glucose levels stable so that they're stable for the brain. And in effect, what you've done is just shunt glucose from non-thermogenic tissues to thermogenic tissues using this adjustment of insulin secretion and insulin sensitivity among the different organs. And then a final thing that happens that's also been discovered upon cold exposure fairly rapidly is an activation of a specific type of neuron that's now well studied in the brain called AGRP neurons. These are the hunger or satiety neurons, and these become rapidly activated also by cold exposure far before there's any change in body weight or any change in energy balance. But these become activated in the cold, and that provides increased food intake and increased energy intake to match the energy expenditure of this of shivering and BAT thermogenesis. So you can see how these three different homeostatic systems are all engaged, and the coordinator of this is really the nervous system. And so it's engaging each of these different systems, and it's doing it in a more prospective, anticipatory way, rather than waiting for these metabolic variables to dramatically change before some negative feedback signal is able to elicit these. Because after all, we want to keep these variables homeostatic. We want to keep them stable. That's the whole point. So now to kind of simplify it, and I'm going to go through each of these. So we have the brain, and then there's outputs in magenta, and then in the blue we have the inputs, and the outputs go to effectors, like the peripheral organs I talked about, to change the glucose level, and then the input comes from sensors in various places to feed back to the brain. So starting with the outputs, and in an effort to try to make this more relevant, or something that we're all more familiar with in terms of how the brain may be involved in glucose homeostasis, everyone's pretty comfortable with the fact that the brain's important for counter-regulatory responses. So in the setting of hypoglycemia, the sympathoadrenal responses that help to elevate glucose levels back to normal, there's longstanding evidence that the nervous system is important for that. And what I'm going to try to do as a theme through these different components is to show you how those same systems that are important for counterregulation are also used physiologically day-to-day, moment-to-moment. It's just that counterregulation represents one extreme kind of version of that in the setting of hypoglycemia. So now these are studies done by the late Dan Port in his lab long ago, but they provide really nice illustration. These are human patients. So what they're doing here is an IV glucose tolerance test, and you can see that in normal subjects, you give an IV glucose load and the glucose levels go up and then they come back down. Then what they did is they infused phentolamine, which is an alpha agonist to suppress, to basically mimic sympathetic tone, an alpha antagonist to block sympathetic tone to the islet. And in the setting of that alpha agonist, you see that first there's a significant, albeit slight, increase in basal secretion.
Maybe.
Oh yeah. Yeah. So there's a significant but subtle increase you can see in basal secretion, and there's also a significant rise in the glucose-stimulated insulin secretion, and that's in normal subjects. And then this is a graph we're all familiar with, which is that in an IV glucose tolerance test, you have normal insulin secretion, and then diabetics don't have this secretion. So then they ran the same study in diabetic patients, and what you see is that in this IV glucose tolerance test, again, you have this small little bump in insulin secretion, but when you give the alpha antagonist to block sympathetic tone to the islet, you get an increase in basal secretion, so comparing basal secretion to basal secretion, and then you get a 5-fold increase in insulin secretion in response to glucose. So these examples show, one, that there's normally mild constraint of the beta cell just in the basal state with no other, this isn't counterregulation, this is just in the basal state. In the setting of phentolamine, you get an increase in insulin secretion, and then in response to a glucose load, you get an even bigger increase. And furthermore, that in diabetic individuals, this is also present and is present maybe in an exaggerated way. So what about the liver? Just as another example. So the liver is this very uniquely specialized organ in that it's both a glucose producer in the fasted state and a glucose consumer in the fed state. And work by Dr. Alan Sherrington and colleagues over the years has really nicely looked at this transition from the fasted to the fed state to illustrate how the nervous system is involved in this. And something that they've defined is what's called the portal signal. So the portal signal is a gradient between the level of glucose in the portal vein versus what's in the systemic arterial circulation. And what you can see in these experiments, what they did, so these are conscious dogs, and they have catheters, and they're inducing hyperglycemia in a pancreatic clamp. So they've removed the effect of islet hormones, and now we're just going to induce hyperglycemia, and we're going to do it in a couple ways. One is through a peripheral glucose infusion. The other is some of that peripheral infusion that's inducing the hyperglycemia is going to be given directly into the portal vein. The total arterial glycemia is the same, but we're shifting it from a peripheral infusion to partially being a portal infusion. And what's shown on this axis is hepatic glucose balance. So anything on the top side is glucose output, and anything on the bottom is glucose uptake by the liver. And what you can see that happens when you infuse the glucose portally, even though, again, the total level of systemic hyperglycemia is the same, when it's coming in through the portal vein, there's an increase in uptake. If you compare this to what happens when you give insulin here is in the black. So insulin does the same thing. It increases glucose uptake. If you combine the portal infusion with insulin, you see an even more rapid and more precipitous decline to a lower value. So there's something beyond what insulin does at the liver to increase glucose uptake at the liver with, for example, after a meal. Okay, and the portal response is much faster, or maybe we don't need to say much, but the portal response is anyway faster than even the insulin response. Now then they went on to look at, so what is responsible for this portal signal? And so in these experiments, it's kind of a similar setup as these. Here, the hepatic glucose load in the infusion is the same, but they're going to give that load either through a peripheral infusion or at least partially through portal infusion. And when you switch in control animals, if you go from a peripheral infusion to a portal infusion, you can see that hepatic glucose uptake goes up, similar to what's described here. Now, if you take dogs and you denervate the sympathetic nerves going to the liver, you can see that goes away, that delta goes away. So now even with the peripheral glucose infusion, the liver is taking up more glucose. And so what this suggests is that in the basal state, when you haven't had a meal, that the liver is under chronic sympathetic tone that's going to keep it producing glucose, and that a meal with a sudden increase in portal glucose is going to turn the liver into a glucose consumer. And then a final experiment they did is they took high-fat fructose fed dogs. So this is a diet-induced model of obesity and diabetes, and you can see that in the basal, in the intact condition, normal dogs, this is their glucose tolerance curve. These high-fat dogs have glucose intolerance, and when you denervate the sympathetic nerves going to the liver, you can get an improvement in their glucose tolerance, albeit not to normal. Okay, so now moving on to the brain and sort of sticking with this idea of counterregulation. You know, the previous responses I showed you, those are responses important in counterregulation. Over the past 10, 20 years, we've become much more aware of the specific subsets of neurons in the brain that are important for these counterregulatory responses. This was one of the first, if not the first paper to identify a specific subset in a part of the hypothalamus called the ventromedial nucleus. So here's the coronal section, here's the ventromedial nucleus bilaterally, here's the third ventricle. And what they do is they use an optogenetic tool where they can specifically activate just this group of cells and activate or inhibit this group of cells and see what happens. And in this experiment, they did an insulin tolerance test. So this is insulin-induced, trying to induce hypoglycemia with an insulin injection. And when the laser's on, which results in inhibition of this particular cell group, you can see that the animals can't respond normally. So they can't mount an appropriate counterregulatory response, whereas when the laser is off, they mount an appropriate counterregulatory response, suggesting that these neurons are critical for that counterregulatory response. Now, further insights are gained by activating these cells. So when you activate these cells, you get diabetic range hyperglycemia. Okay, so this is, obviously this is an artificial state. We're activating this population selectively. You get diabetic hyperglycemia, but look what happens at the insulin levels. And by the way, you don't get that in controls, and you don't get that in a different population of neurons that's not this specific population. You don't get a compensatory insulin response. So normally, if your glucose levels go up like this, you should get a big insulin response, but you don't. And that again goes to the point that this is a coordinated thing that the brain is doing. That's not something that the islet can compensate for or overcome, because part of this response involves reducing glucose-stimulated insulin secretion. So overactivity of these cells produces that. Now then, this work has been taken a little bit further, looking at an even more selective subset of VMN neurons, which express, it doesn't really matter, but this is the cholecystokinin B receptor. And here is that population of neurons. And if you activate these cells, they produce an even more dramatic hyperglycemia than the previous cells I showed you in the last figure. And what's interesting is, if you inactivate these cells in otherwise normal animals, you get a fall in the basal glycemia. So these are just normal, fasted glucose levels in these mice, or fed glucose levels in these mice. These are control mice. These are a different population of VMN neurons. This is the population of VMN neurons that I'm talking about here, the CCK-BR neurons. If you inhibit these cells in otherwise normal mice, your euglycemic value falls to a lower value than it was previously in both states. So that suggests that under basal normal conditions, this population helps keep euglycemia where euglycemia is, which is slightly higher than where it is in these mice. And then furthermore, they did some other experiments to show that this happens in an insulin-independent fashion. So it's not dependent on insulin, it's dependent on direct control of hepatic glucose production. If you take streptozotocin-induced type 1 diabetic mice, so you give mice streptozotocin, which is a toxin for beta cells, it kills all their beta cells, their insulin levels drop to near zero. And in that setting, you get this diabetic range hyperglycemia, uncontrolled hyperglycemia. If you inhibit these cells in those animals, you get an amelioration of that hyperglycemia, which is even more interesting because it suggests that now an insulin deficient state is actually somehow promoted by this population of neurons in the brain, such that if you take that population out, you can even get improvements in insulin deficient diabetes. We can talk more about that if there's questions, but just in the interest of time, I'm going to move on. So then how does this sensing work? So for counterregulatory responses, the epinephrine response to hypoglycemia is mounted within a minute. So it's a very rapid response. And there's been lots of studies looking at glucose sensing neurons and how neurons in the brain may sense glucose in the interstitium. But the problem is that glucose levels in brain interstitial fluid change very slowly in comparison to glucose levels in the blood. So on the order of 10 to 30 minutes after a change in the blood, you get changes in brain interstitial glucose. So really the way to look at this is in vivo, and studies are now just, with the technology that's available, coming out to look at this in vivo. So what are the responses of neurons in the brain to a changing glucose level? And this is done using a technique called fiber photometry, where you can do calcium imaging of a select subset of neurons. And so this is done, again, on a subset of VMN neurons. That's the theme here. It's not that other neurons aren't important, just these are the neurons I'm happening to focus on for this talk to kind of provide a theme so that you can kind of see it all the way through. But this subset of VMN neurons has been recorded from in the setting of a glucose tolerance test. And what you see is that very quickly as glucose levels go up, the activity of these neurons falls. And if you correlate the change in glucose to the change in neural activity, this is a cross-correlogram, they're maximally negatively correlated with an offset of 60 seconds, where the blood glucose level precedes the change in neural activity by 60 seconds. And if you look at a histogram with a 60-second lag between this signal and this signal, this correlation is maximized at that minus 60-second lag. So here's an example that actually fits what happens physiologically in vivo, which is that these neurons are rapidly activated. By changing glycemia. And this is my last slide. So in this, the final thing is, so how does this information get to those neurons? And this is work done by Alan Watts and Casey Donovan. And so here, these are, again, a dog model. They're looking at the response to systemic hypoglycemia. And so here's insulin. They raise the insulin level, and then they do a glucose infusion to slowly induce hypoglycemia. And what they're showing here is that between their control and their experimental conditions, there's really no difference in insulin levels, no difference in arterial glycemia or the glucose infusion rate. The only thing they've done is that in the portal infusion animals, the glucose is going in through the portal vein. In the peripheral infusion animals, it's going through the peripheral vein. And when you give the glucose infusion through the portal vein, basically tricking the liver that the glucose levels are higher than they are and producing that gradient, you don't get the counterregulatory response. So you can see the animals that have the portal infusion, they don't mount the counterregulatory response, despite the fact that they have systemic hypoglycemia, because the sensors in the liver think that everything's okay. And then they went on to show that this is actually mediated by spinal sensory nerves that express a particular channel that's taken out by capsaicin. So these are now rats. Capsaicin is painted onto their livers that ablates these nerves. So here's the control nerves, over the portal vein. Here's the denervated animals. And in the denervated animals, you can see that animals that don't have those spinal sensory afferents over the portal vein don't mount an appropriate counterregulatory response. So the summary of this kind of broad overview is that the brain integrates sensory signals of circulating glucose levels. It's important that the brain nervous system as a whole is involved in this because it's also able to integrate this with interceptive signals of metabolic state, what's happening in the environment, what are my planned behaviors that might need glucose, and then provide descending control both of insulin-dependent mechanisms, but also insulin-independent mechanisms. Thanks for your attention.
We have time for one quick question.
Blandine Laferrère, Columbia University, New York. A great talk, really, very nice. Quick question, could you give a word on the role, perhaps, of the central nervous system on the absorption of glucose in the gut, if any, or the reabsorption of glucose at the kidney level?
Oh, I'm sorry, I can't, but that's very interesting. I don't know if you guys have any. Yeah, that's a really interesting question. Obviously, the gut is, very densely innervated. Yeah, I'm sorry, I don't have anything to add to that.
Thank you very much.
Yeah, thanks.
Great. So our next speaker is Dr. Kevin Williams. He is an associate professor of internal medicine at the University of Texas Southwestern Medical Center in the Center for Hypothalamic Research. Dr. Williams and his team investigate the underlying physiology of how neural circuits and signaling mechanisms in the brain regulate feeding behavior, energy expenditure, and glucose metabolism. Today, he's speaking to us about the role of exercise to improve metabolism via action within the brain.
Should I get this? Okay. I think we're good.
We're good?
Yep. So thanks for the invitation to come speak today, and thank you for showing up so early in the morning. I've been talking about the plasticity of neurons in the brain in response to exercise, with a particular focus of the hypothalamus and even more specifically in the arcuate nucleus. Does this work?
It should. Are you looking for the laser or the?
No, the advancer. No.
Maybe not. He was using it.
Let's see how this goes. Okay. So I did say we're going to talk in the arcuate nucleus, talk about the arcuate nucleus plasticity. I don't want to dismiss the complexity of this circuit or the circuits in the brain that are regulating metabolic homeostasis. The arcuate nucleus, as I'm going to point out later, is down here in the, oh, it doesn't show up on there. There we go. In the base of the brain. Right there in the center, you can see other hypothalamic nuclei and cell populations. These cell populations can be identified by different receptors that are expressed there. These receptors can be different humoral cues coming from the periphery, but they can also be receptors to peptides and neurotransmitters within the brain. These neurons can also be identified by different proteins that they're expressing. So it's different neuropeptides, neurotransmitters that they express, and also their projection patterns. You can see that these hypothalamic nuclei and cell populations are projecting all over the brain, down into the hindbrain, the spinal cord, up into the cortex, and they commonly receive reciprocal projections back from these downstream/upstream neurons and nuclei. So it really is the coordinated activity effort of these neurons and nuclei that's important in maintaining metabolic homeostasis. So there's the arcuate nucleus, and we're interested in this area of the brain because of two cell populations, the MPY-AGRP, or the neuropeptide Y-agouti-related peptide neurons, as well as the propiomelaanocortin and POMC neurons. And we're interested in these neurons because there's a lot of classical literature from lesion studies, molecular genetics, pharmacology, optogenetics, chemogenetics, which has shown that these neurons are important in the regulation of feeding behavior. They're also important in the other side of the energy balance equation, regulating energy expenditure through classical thermogenesis of brown adipose tissue, as well as browning and beiging of white adipose tissue. They're also involved in the regulation of glucose and production and utilization in a variety of tissues. Now, importantly, the activity of these cells, and Zeman nicely introduced some optogenetic data. Chemogenetics has also been used in these cell populations in order to drive or inhibit the activity of these cells, and it's been shown that if you activate MPY-AGRP cells and or if you inhibit POMC cells, this typically will result in an increase in feeding, a decrease in energy expenditure, and an impairment in glucose metabolism. The opposite activity, to inhibit MPY-AGRP cells or activate POMC cells, can typically result in a decrease in food intake, an increase in energy expenditure, and an improvement in glucose metabolism, which can be read out as improvements in insulin sensitivity. Now, these neurons are highly plastic. They're highly malleable to a variety of conditions. This can include different types of metabolic cues from humoral signals such as leptin, GLP-1, and ghrelin. It can also be different types of metabolic challenges such as food deprivation or high-fat diet feeding. Now, we know that exercise is also a metabolic challenge. Single bouts of exercise or chronic training can improve metabolism. In the periphery, this can result in improved cardiac function, an increase in insulin sensitivity, improvements in physical fitness and fatty acid mobilization. Within the brain, this can result in neurogenesis, synaptogenesis, and plasticity. In studies of exercise-induced plasticity in the brain, this has largely been limited to studies within the hippocampus, looking at long-term potentiation, learning and memory, cognition. Very few studies have actually looked at metabolically relevant cell populations and the plasticity of those in response to exercise. So we wanted to ask a simple question: if some of these metabolically relevant cell populations could be plastic in response to exercise, and in particular, if exercise could cause changes in activity of MPY-AGRP and POMC cells, and what that might mean for metabolism. In order to do this, we performed a high-intensity interval exercise, so 1 hour HIIE, and this is a ramping of speeds in our rodent models on a treadmill, so it's forced exercise. We go from 12 to 22 meters per minute, and we repeat that 3 times for 1 hour. We also did a chronic exercise training over 5 days, as well as a 10-day exercise protocol. After that exercise protocol, we immediately did whole cell patch clamp electrophysiological recordings, or we did fibrophotometry. Now, this is a schematic of the fibrophotometry recording, in which we have a mouse on a treadmill. It's tethered to a fiber optic probe for the fibrophotometry recordings. We used the AGRP Cree and the POMC Cree mice, and we injected those with the AAV GCAM6S, which is a calcium sensor. So this calcium sensor, when calcium levels go up, you get more fluorescence. If calcium levels go down, you get a decrease in fluorescence. The green signal is the calcium sensitive signal. The purple signal is a calcium independent signal. So that's the isosbestic signal. And so what's shown here is 10 minutes before the end of the run and 10 minutes after the run. And we have our AGRP neurons on top, the POMC neurons on the bottom. You can see a decrease in the calcium signal for the AGRP neurons and an increase in the calcium signal for the POMC neurons. This would agree with an increased activation of POMC neurons and an inhibition of MPY-AGRP neurons. Now, we did confirm these data post-exercise with whole cell patch clamp electrophysiological recordings. However, for time constraints, I'm not showing that data, so I'm going to summarize the work here, in which post-exercise we saw an increase in activity of POMC neurons and an inhibition of MPY-AGRP cells. We also were interested in the synaptic inputs to these cells, and after exercise we saw that the POMC neurons, it's actually immediately 1 hour after the 1 hour exercise bout, we had an increase in excitatory synaptic strength to the POMC cells that actually persisted, potentially became stronger with chronic exercise training. The MPY-AGRP cells saw an increase in inhibitory synaptic strength, not immediately after the single bout of exercise, but after chronic training. This could suggest some plasticity that's occurring in this circuit in response to exercise. So in addition to looking at single time points right after an exercise bout, we're also interested in how long these effects last and when they might be reversible. And I should point out, going back to some of the plasticity of this circuit, there is a history to looking at the plastic nature of these neurons. And this is data from Jeffrey Friedman's group and Tomas Horvath, in which they looked at the plasticity of these circuits from wild-type mice to OB mice. And what you can see is that the MPY-AGRP cells receive a larger excitatory tone in the OB mice compared to the wild-type condition. The POMC neurons receive a larger inhibitory tone. And as many of y'all are aware, the OB mice, similar to humans that are leptin deficient, they're hyperphagic, obese, and partly diabetic. The activity profile of these neurons is in agreement with that physiology. Now, what was remarkable is that if you gave leptin as a monotherapy, if you treated the deficiency, the circuit could rapidly rewire back to its wild-type state. And this could occur within 6 hours. Another plasticity of this circuit is in response to food deprivation. So fasting overnight can rapidly activate MPY-GRP cells. And this data is, from Roger Cohn's early work, the first description of this, in which MPY-GRP cells could be activated in response to overnight fasting. Multiple other investigators have confirmed this, and they've suggested some calcium signals can be involved in this process. Specifically, Brad Lowell's group showed that this fasting-induced activation of MPY-GRP neurons could last for about 3 days after that single overnight fast, and that NMDA receptors were required for that activity. As it relates to the physiology of exercise, single bouts of exercise can result in an improvement in insulin sensitivity that can last for 24 to 48 hours after that single bout of exercise, and that can occur across multiple species from rodents to humans. So we had several time points to investigate the reversibility of the activity in our circuit. And what's shown here is we're looking at the resting membrane potential, resting membrane potential of POMC and MPY-GRP cells from mice that were sedentary or they experienced the 1 hour exercise. The black line going across the screen from left to right is the resting membrane potential of POMC neurons or MPY neurons from sedentary mice. The red is after a single bout of exercise. So you can see that the POMC neurons have a positive deflection in their membrane potential or a depolarization. The MPY neurons have a negative deflection in their membrane potential or a hyperpolarization. So their POMC neurons are activated, the MPY neurons are inhibited. If we look 24 hours after a single exercise bout or 1 day later, the POMC neurons are still activated. They remain activated for 2 to 3 days after that single bout. If we look at the MPY neurons 24 hours later or 1 day later, they already returned to their sedentary state. 1 hour after the single bout of exercise, they might be more inhibited, but they return to that sedentary activity profile within that 1 or 2 to 6 hour window. So we have a temporally distinct activity profile of the POMC and MPY-GRP neurons, in which the MPY activity reversed within that 2 to 6 hours, and the POMC neurons reverses within about 3 days. So what can this all mean? Well, this activity profile has been associated with a decrease in feeding behavior. Changes in feeding in response to exercise can be quite controversial, but high-intensity exercise can typically result in decreases in a drive to eat, and this is some data from young Caucasian males running a 75% maximum oxygen uptake on a treadmill. They're asked to report their hunger scores. During the run, they have a decrease in their drive for hunger, and that persisted for the first hour or two after that exercise bout. We did a similar experiment in our rodents, and after the single bout of exercise, there was a flatlining in the feeding behavior that occurred within that first 1 to 2 hours, but then the slope of the line of the mice that had been exercised started to catch back up with the sedentary mice, suggesting that there was a slight suppression or a transient suppression in the feeding behavior after the exercise. Now, due to the activity profiles of the MPY-GRP neurons reversing within about 2 to 6 hours after the exercise bout, we would suggest or posit that maybe that activity might contribute to this, but we don't have any direct evidence of that. This activity profile is also in agreement with an improvement in insulin sensitivity or an improvement in glucose metabolism. And we were very interested in trying to further investigate this, and POMC neurons have been suggested. If you drive the activity of them, they could improve insulin sensitivity. We noted that the POMC neurons were activated at the same time they were receiving a larger excitatory input, so they were receiving a large glutamatergic input into these neurons. And we noted that this was very similar to what was happening in the MPY-GRP cells after an overnight fast. I mentioned earlier, Brad's group had suggested that an increase in excitatory input driven by NMDA receptors was required for this activity. We wondered if a similar mechanism could be taking place in POMC neurons following exercise. In order to do this, we obtained a Cre-inducible mouse model of POMC Cre-ER, cross that to the NR1 subunit, deleting out NMDA receptors and POMC neurons. And when we did this, we have our POMC neurons from mice that are deficient from NMDA receptors, and we exercise them. And so this is sedentary mice that are deficient for NMDA receptors and POMC neurons. And you can see in the exercise condition, there's no depolarization. We don't see an increase in the firing rate. We don't see an increase in excitatory input to these neurons, which is what we observed in the wild-type mice. So we were able to abrogate the exercise-induced activation of POMC neurons. So what does this mean physiologically? We performed an insulin tolerance test after our exercise bout, and in this condition, we performed a 1-hour HIE and then 3 hours of food deprivation, so for a 4-hour total food deprivation before the insulin tolerance test. And what's shown here is the wild-type mice from the sedentary mice and then the mice that had the exercise bout. And you can see that the baseline blood glucose levels are elevated after the exercise bout, but after exercise, the mice have a larger drop in the blood glucose in response to insulin. Now, we also noted these mice were going through a slight hypoglycemic bout in response to insulin, and we wanted to avoid interpretation of the counter-regulatory mechanisms. And so in order to do that, we focused on the first 15 minutes of this insulin tolerance test. And you can see that there's the drop in the blood glucose that occurs within 15 minutes. We performed the same ITT 24 hours after the exercise bout. We see a similar drop in the blood glucose levels, suggesting an improvement in insulin sensitivity. And this actually persisted 48 hours later, but was starting to subside. If we looked at our mice that were deficient for NMDA receptors and POMC neurons, they do exhibit an improvement in insulin sensitivity. However, overall, this is largely abrogated. So mechanisms involved in the activation of POMC neurons, inhibition of MPY neurons. I think I've shown that the NMDA receptors might be at least partly involved in the exercise-induced improvements in insulin sensitivity. We don't quite know the mechanism that's taking place in the MPY-AGRP cells. But we need to pay attention to the synaptic inputs as well as the outputs of these cells. It might be relevant in the immediate effects that are observed after a 1-hour exercise bout, but it could also be relevant to more chronic exercise training. And this takes us back to this diagram in which all of these cells are interconnected with reciprocal connections to different types of target organs. So downstream neurons could be upstream neurons, and we're showing a drive in the activity of POMC neurons that appears to be dependent on some other neuron in a different location. There can be questions about forced versus voluntary exercise, and this could be answered in part by some work by Tomas Horvath, in which they performed fibrophotometry recordings on mice in which they recorded the AGRP activity. And they did this on mice that were running on a treadmill voluntarily. And so what they show is this is 30 seconds before the beginning of a running bout. This is 30 seconds after the beginning of a running bout, 30 seconds before the end of the running bout, and 30 seconds after the running bout. So you can see at the beginning, they're not seeing any changes in the calcium signal. But at the end of the running bout, they're getting a decrease in calcium, suggesting that the cells are inhibited at the end of exercise. But you might notice, or if you paid attention, that this is very transient. It's reversing out within tens of seconds after that single exercise bout. There could be some, it does mirror our effects in forced exercise, but there could be some explanation for these differences. One of them could be the potential duration of the exercise bout. Since the mice are undergoing voluntary exercise, many of the exercise bouts that they ran were somewhere between 100 to 300 seconds. On our forced exercise, we're running mice for up to 1 hour. So possibly the duration of exercise might contribute to how long the AGRP neurons would be inhibited. Another contributing factor could be the speed. Since they're voluntarily running, they're not running at the high intensity level that we ended up forcing our mice to run. So duration or speed could contribute to some of the differences observed. Temperature could also be a potential mechanism in this process. Many of us are aware that you could raise your body temperature in response to exercise. This is work from rodents in which measuring rectal temperature and also temperature within the arcuate nucleus. And they're seeing a rise in body temperature as well as temperature within the hypothalamus in response to an exercise bout. This is also interesting. Get back over here. Because they show an exercise-induced hypophagia in response to their exercise bout when they administered the SHU compound, which is an MC4 receptor antagonist. They can abrogate this exercise-induced hypophagia. MC4 receptors are downstream of POMC and MPY-AGRP cells, so it could suggest a role of POMC and or AGRP in this process. There's a variety of nutrient sensor signals that can be contributing to changes in this circuit. There's some other potential exerkines that could be going into the brain to cause plasticity or changes in activity. It's unclear right now if one of those could be the drivers. There is some recent interest in lactate and lactate metabolites and potentially driving changes in plasticity. Here's a paper from this last year suggesting that lactate metabolism within the arcuate nucleus could be contributing to exercise-induced effects, including exercise-induced changes in feeding behavior. They did observe at the same time a decrease in MPY protein expression that lasted for about 6 hours, an increase in protein expression for POMC that lasted greater than 24 hours. This was very analogous to the changes in the activity profiles that we were observing in the POMC and the MPY cells after exercise, but we're not quite clear if this could be a potential driver. There could also be sex or circadian dependent effects that we need to be paying attention to. All the data I showed you were from male lean mice. We haven't examined what's going on in different types of disease states. So it'd be very interesting to better understand what's going on in diabetic models as well as DIO models or obese conditions to see if a similar activity profile might be observed to contribute to improvements in metabolism. And while it's generally accepted that exercising at any time of the day whenever you can is a good thing, there is some recent data suggesting that there could be time-dependent improvements in metabolism in response to exercise. So you might be able to enhance the effects as it relates to insulin sensitivity or maybe even energy balance changes. And so coming back to this model where these neurons are highly plastic and malleable to different types of hormones and metabolic challenges, we think that exercise could be added to this list of metabolic challenges that can induce a plasticity of this circuit, resulting in some type of a hypothalamic LTP that can improve some kind of a metabolic state. This could ultimately affect all these other cell populations, but our data also suggests that these other cell populations are influencing this hypothalamic LTP. And we really want to try and identify what this factor is or factors that could be contributing to these changes that is quite unclear right now. And so acknowledging people that did the work, most of my lab has been comprised of technicians. Unsong Huang is a postdoc, Yanbin Dong is a student, Lynn, Brian, Brianna, Anita, Jason, Kyle are all technicians that have contributed to each various components of this study. I've benefited from multiple collaborators at UT Southwestern, as well as outside of UT Southwestern, so Joel Elmquist, Jeff Sigmund, Phil Scher, Nick Betley, and Amber El Haydef. And then some shameless self-promotion. For this past year, I've been co-hosting the Diabetes Bio podcast with Darlene Sandoval and Kirk Habegger. We've been doing it just this year. It's the official podcast for the Journal of Diabetes. We try and highlight particular papers in each issue, so it's a monthly podcast, but we're also highlighting special topics, anything from interviewing award winners, so ADA Pathway Award winners at the meeting, also special kind of debate issues, point-counterpoint topics, and so I would encourage you guys to check it out. I think it's been very informative and quite interesting. We are actually meeting tomorrow in the exhibit hall at about 12:30 to 1:30 with the other podcast team of the American Diabetes Association. We are just the babies of the podcast team. The other teams have been around for a few years. Tomorrow will include Alice, Mike, and Neil. And so please come check us out, talk to us, give us some feedback. We'd love to hear from you. And I wanted to give a special recognition to Dave Wasserman. We ended up losing him this last week. He was very vital in getting our exercise project up and running. I got to know him over maybe the last 7 to 10 years, and he's been a great mentor, colleague, and friend. And so he's gone too soon. And so thank you.
Thank you so much. We have time for one quick question, if anyone has one. So I'm particularly fascinated by the fact that, like, after you exercise, particularly mice, they won't eat for about an hour, right?
Uh-huh.
You're saying?
Yeah.
So is that, like, reversible if you stop the neuronal, like, signals down? Like, would they...
So a de-innervation or
or
like
POMC knockout
or
or AGRP
a
knockout.
chemogenetic manipulation. So we've thought about that. So the chemogenetic inhibition or optogenetic inhibition of MPY-GRP cells is quite strong and profound. You will have a change in feeding behavior immediately with those types of manipulations. And so trying to control for that kind of caveat, to where you're not just blocking a signal. And so the potential way we could do that is maybe blocking synaptic transmission coming from those neurons. So there's some strategies of like using a TET toxin or something like that in order to just block that synaptic transmission. But we haven't done that type of study.
And what would be the physiological consequence if the mice or humans started to eat right after exercise? Like, do you think the glucose regulation would be affected the most, or?
It's a good question. I don't know. I've thought about why. Why are they shutting off feeding behavior? And rodents are they're not animals that exercise per se, but it could be a fight or flight type response, trying to avoid a predator, and it's not a time when you need to have driving of a food-seeking drive. So it could be a situation like that. There's been some evidence, which I put up on the screen. I didn't really go into it, but driving the activity of MPY-AGRP cells can shift substrate utilization to carbohydrates. Okay. It hasn't been really evidence to suggest that shutting it down would shift it to utilization of fat, but it could be a way of utilization of substrate stores during physical activity. So it could be something related to that.
Good. Thanks so much.
Mm-hmm.
So last but not least is Dr. Christy Townsend. She's an associate professor in the Department of Neurological Surgery at The Ohio State University and runs the Neurobiology and Energy Balance Lab, focused on investigating how the brain and peripheral nerves impact metabolic health. Dr. Townsend is also the director of Women in Medicine and Science.
All right. Well, thank you to the organizers. This session was called It's All in Your Head, but I think as we're seeing today, a lot of it's not in our head and it's in the peripheral nervous system. I'll be sharing some published and unpublished data today, so I just ask that nobody please photograph the unpublished slides. So we already had it introduced what the peripheral nervous system is, but I'll just briefly review that we've got efferent nerves that are signaling from the brain out to the tissues, as well as the afferent nerves that signal from the tissue back to the brain. And these can be vagal or spinal, depending on the tissue and organ that we're talking about. And if you've ever learned about the peripheral nervous system in a class previously, you might see a diagram like this showing the gut and the liver, as we've talked about today, but you never see that the adipose tissue is one of these tissues innervated by the peripheral nervous system. But in fact, adipose tissue as an organ is very complex. It's got a blood and lymphatic supply and a rich nerve supply from the sympathetic nerves and the sensory nerves. And this tissue has many functions, including I got to click differently, including endocrine, metabolic, and immune roles. And when there are changes in energy balance, the tissue can really rapidly remodel in response to that need. Historically, most of the work has been done on the sympathetic nerves that innervate adipose, but we now know that there's a nice sensory nerve supply, including those that have calcitonin gene-related peptide or CGRP. And I just want to note here, for people used to seeing these tyrosine hydroxylase nerves in the tissue that are typically called sympathetic, actually 40% of those are sensory and have sensory neuropeptides. So just something to keep in mind. But in general, as we're getting signals from the brain or to the brain, those interact with the peripheral nervous system and release neurotransmitter and neuropeptide into the tissues and organs, either from their terminals or, as I'll show you today, they can also release en passant. And in adipose particularly, there are many functions that these nerve products have on the adipocytes that are important for metabolic health. So these nerve products can control adipogenesis, lipolysis, and browning or beiging. And a lot of work over previous decades using surgical, chemical, genetic, or viral methods to denervate different nerve subtypes have shown that both sensory and sympathetic can contribute to these processes. So the sensory nerves are a little bit unique. They're afferent. Their cell bodies are up in the dorsal root ganglia, and they're releasing nerve product like neuropeptides out into the tissue, but the electrical signal is going back up to the brain, including to the hypothalamus. So what you end up with is a bidirectional neural circuit where you've got efferent signaling, things like norepinephrine coming down to the tissue, and then afferent electrical signaling back to the brain and release of neuropeptides into the tissue. So we know that specifically these sensory nerves in adipose control lots of different things, lipolysis, thermogenesis, now vascular control as we're seeing. through the release of their neuropeptides and as well as that electrical signal going back up to the hypothalamus. But there's still a lot we don't know. So our lab is looking at some of these open questions like what are the interoceptive signals coming from the tissue? How are these neuropeptides acting on the different cell types? And how do the sympathetic and sensory nerves crosstalk? Because they definitely do. But I won't have much time to talk about this today. And instead, I want to focus on something really relevant for diabetes, which is peripheral neuropathy. So this is a neurodegeneration of the peripheral nerves. It's actually the most common neurodegenerative disease in humans, and it results in a loss of tissue and organ innervation and blunted neural communication with the brain. With diabetes, this is often length dependent, and it's caused by over 30 medical conditions, but diabetes is by far the number one. Some more recent data showing up to 70% potentially will eventually develop neuropathy, and it's detectable in prediabetes and obesity as well. It's typically a polyneuropathy, so the symptoms are complex. If left untreated, it can lead to limb amputation. And it's typically measured by skin sensation or innervation density tests or looking at large fiber nerve activity. But there's lots of evidence that probably multiple tissues and organs are impacted, and we're really not able to detect those other tissue and organ neuropathies in the clinic. I just want to direct everybody to this great review that just came out by my colleagues at Michigan talking specifically about obesity-related neuropathy as well. So in our lab, we've been focused on the neuropathy that can happen to our adipose tissues, which we call adipose neuropathy. And I'm just summarizing what we have now for data across multiple studies. But essentially, if you look at the diagram in a healthy tissue, you should have innervation at your skin surface, subdermally, and down to your subcutaneous adipose. But when the neuropathy starts, you get denervation of the skin, and then later in disease, denervation of deeper tissue layers, including adipose and muscle. And we've shown this through lots of different tests now, looking at nerve, synaptic, and growth cone markers. There's reductions in neurotrophic factors in the tissue. We now can measure that there's altered electrical activity of these transdermal nerves. And I'll show you today some of the work on demyelination in Schwann cells. So because, as I showed you, that when we experimentally remove the innervation to adipose through those denervation techniques, that dysregulates adipose tissue functions. So we think when we undergo adipose neuropathy, this could also exacerbate metabolic disease by reducing that important bidirectional neural communication with the brain. And we have seen adipose neuropathy in human and mouse samples with obesity, diabetes, and also aging, which I'll talk a little bit about today. So this is one example of the data that we see in adipose where it's really a small fiber neuropathy. So the small diameter axons are lost, just like in the skin. So you can see in the wild type animal on the left, in between the large nerve fibers, there's lots of small fiber innervation. And this is an example from an OBOB mutant that there's loss of the small fiber axons. So another thing we see is that there's demyelination and alterations to Schwann cells when there's adipose neuropathy. So first, to embark on this study, we had to demonstrate that there actually were Schwann cells in the tissue and actually myelinated nerves in the tissue. So both are there. So on the left is an example of a Schwann cell that's embedded along the myelin sheath of an axon, and Schwann cells, as a reminder, are the myelinating glia of the peripheral nervous system. And by electron microscopy, we could see both myelinated axons and thinly myelinated or unmyelinated axons in the tissue. So when we have an obese diabetic model, this is the OBOB mouse, we have a reduction in innervation in the tissue that we can show by Western blot and also by imaging. And this coincides with a reduction in the total number of Schwann cells in the tissue and also the total amount of myelin in the tissue. And we can also image this and see the demyelination and the axonal degradation. So on this image on the top, this is a wild type axon in green, and then the myelin sheath around it in red. And then the mutant on the bottom, you can see the loss of myelin and also the fractionation of the axon. So we think this adipose neuropathy is also a demyelinating neuropathy with obesity and diabetes. And now Jillian Gunch in the lab is working on this more mechanistically, and we're looking at what role the Schwann cells actually are playing. Why are they not repairing these nerves when they really will do that in a nerve injury in the large fibers? So we're looking more at their mechanistic involvement. So just to summarize, in adipose, we have large fiber nerves. We have thin fiber, small fiber innervation in between that. We also have a lot of neurovascular interactions. So here's an example of the sympathetic fibers around the blood vasculature. And then we have myelinated axons and Schwann cells. But what's interesting is when we look at the pattern of presynaptic proteins in the tissue. So this would indicate that there's some sort of synaptic terminal in that region. So this is one of the synaptic proteins, synapsin 1 in red across the subcutaneous adipose depot. And I've overlaid below it Paul Cohen's clearing of the same depot showing sympathetic innervation in purple and UCP1 in green. And what you'll notice is there's two different patterns here. So there's areas with not much synaptic protein expression on the top, and then areas with very dense synaptic expression. And that really coincides where we have more of the small fiber innervation, more presynaptic markers, and more browning are all in the same regions of the tissue. So we've really been interested in whether there is a neuromuscular junction or some type of true synapse or terminal in adipose tissue, because that had never been described before. But we do think the majority of neurotransmission in the tissue is happening at these varicosities. So these sort of bumps on the axons that you can see here with the synaptic and synaptic vesicle markers in red. And this is similar to what we know exists like in the sheep intestine, where we have all these autonomic varicosities, and this is where the synaptic vesicles are located, and they can release nerve product en passant, basically along the whole length of the axon. So in adipose, we do think a significant amount of nerve product release is happening along the axon, where the products can just diffuse to nearby adipocytes. So if the tissue has neuropathy, cells are further away from these varicosities and not seeing as much nerve product. But we also did identify a unique synaptic structure in the tissue that we call the neuroadipose nexus. And these are nerve terminals where the nerve endings are wrapping around a subset of individual adipocytes. And you can see that here in the imaging. And these have a lot of structural similarities with the neuromuscular junction. So there are myelinated axons and Schwann cells and neuroimmune cells all clustering in these regions. And it turned out when we were writing a review article a few years ago and went back into the historic literature, this had actually been described and drawn by hand by Dogiel, who was a prominent anatomist in the late 1800s. And if you look in the middle of this diagram here, and then above that, he is showing that there's these NANs, these neuroadipose nexus structures around the adipocytes. And this was perivascular or pericardial adipose from a cat. So in total, there's 3 patterns of nerve innervation in adipose tissue. There's a lot, probably the majority is neurovascular around the blood and the lymphatic vessels. There is parenchymal innervation with those varicosities, and then there's these specialized NAN structures that we think are a specialized nerve terminal. So Jake Willows in my lab has been doing a lot of work. He's very interested in these NANs and what they might be doing. So here's what we know so far. So we know that they're very infrequent in healthy and lean adipose. They're mostly at the surface of the tissue. When he looks in all the different mouse white fat depots, they're in every single depot. We're looking in human now. They express synaptic vesicles and synaptic proteins, so indicating they are a true synapse. They have both types of nerve fibers, so we can see TH that are sensory marker negative, so those are sympathetic fibers, and also those that express CGRP that are sensory. And these NANs express TrkA, so they can respond to NGF, as I'll show you in a second. We don't know what's special about these adipocytes that they wrap. Probably there's something unique about them, or they could be representative of the tissue. And we don't know their functions. We have several hypotheses that they could be sensing cell size or mechanics. We're looking at piezo expression, for example. They could be sensing lipids. We're doing a lot of work in that space. They could be nociceptive because these sensory axons are nociceptors, et cetera. But here's what we know about their frequency and how they change in response to energy status. So when we look at high-fat diet, we see a huge increase in the number of NANs across the tissue. And remember, this is a neuropathic tissue, so there's loss of small fiber innervation, but the number of NANs are going up. When we look at aging, middle-aged animals, which is when the neuropathy onset happens in the mice, have an increase in NANs as well. And then lastly, some of the proof of concept gene therapy work we were doing, we were delivering NGF to the adipocytes to help recover the adipose neuropathy. And this was a really effective tool to increase the number of NANs in the tissue, and it also increased the density of the NANs, so how many axons seem to be wrapping an individual adipocyte. And then lastly, we looked at cold because that's really one of the best tools to increase innervation of the tissue instead of reducing it with neuropathy. Cold really increases the branching, especially of the sympathetic fibers or TH positive fibers, as you can see here. But there were no significant changes in the NANs under this condition. And if anything, maybe a slight decrease. So in total, there's a clear disconnect between the innervation density of adipose and the NAN numbers, as we're seeing across multiple studies. Okay, last I'm just going to show a little bit of recent data on our projects looking at aging, because as we know, aging is the biggest risk factor for later disease, including diabetes. Aging has numerous negative impacts on adipose tissue with redistribution and ectopic lipids, chronic inflammation of the adipose depot, and it also has numerous negative impacts on the nervous system. So age-related peripheral neuropathy, denervation of the neuromuscular junction, and as we've shown, adipose neuropathy as well. So we've been working for many years with Jackson Lab and David Harrison, who's part of the intervention testing program. They use these heterogeneous Het3 mice to study longevity treatments, like a couple I mentioned today, rapamycin and estradiol, that can increase lifespan. But very few of these studies have looked at health span in the Het3 mice, so we've been very interested in what's happening to metabolic health, to adipose tissue function, and to peripheral nervous system health. So these are the different longevity treatments that the ITP has been testing over many years across 3 different sites. And they're really interested in lifespan. And only 3 of these have been successful at increasing lifespan in both male and female animals. And that's rapamycin, acarbose, and glycine. So we started with rapamycin in our studies several years ago because we know this has metabolic impacts and wondering if it could improve age-related neuropathy and adipose functions. Whoops. And we actually found that it did not. So neuropathy was not changed at all. And in the animals that were treated either starting at midlife or at late life with rapamycin in the diet actually had much more unhealthy adipose tissue. It was very fibrotic and very inflamed. But what was interesting coming out of this study is we could see that the female mice at middle age were protected from neuropathy versus the males until they went through reproductive senescence or mouse menopause, and then they had much worse neuropathy compared to the males at late life. So then we went back to the ITP list and we said, well, what about estradiol? That was one of the longevity treatments that only increased lifespan in the males, but nobody had looked at health span. And they were using 17-alpha estradiol, which is non-feminizing, so it's a treatment you can give to both male and female mice. So this is what Magda in the lab has been working on. I'm showing some of her preliminary data today, but we're following the same ITP paradigm where we give the animals estradiol in the diet starting either at midlife or at late life. And we can see by mass spec that there's a significant increase at both interventions in 17-alpha in the circulation. And the other, I'm not showing it here, but the other reproductive hormones are not changed. And what was really exciting for us is that the skin neuropathy was completely improved in both sexes. Showing here just the late intervention data, but we're seeing it now in the early intervention as well. So for both sexes here, control is on the top and then treated is on the bottom. And there's just a really robust increase in the nerve fiber density in both sexes treated with 17-alpha estradiol. We also see improvements to adipose tissue, so just showing here the fat mass reduction that we see in both sexes. Glucose tolerance is improved. We're doing a bunch of other metabolic and adipose studies as well. And with Dave Arnold, we've been characterizing the neuromuscular function, and we do see improvements in the neuromuscular function as well, and some other peripheral nerve tests that I don't have time to show today. So a lot more is underway for this, but we're excited about it as a potential therapy for peripheral neuropathy and potentially linking estrogen receptor signaling on nerves to metabolic improvements. So I'll stop there and thank the group. Magda, Jake, and Jillian contributed data to today's talk, and thank you for your attention.
Question in the
Hi,
back.
Christy. Devashish from Ohio State.
Oh.
Nice to see you here.
Hi.
My question is on your the interaction between the sensory and sympathetic. So I would like to know what are your views on that.
Yeah, so we think there's definitely crosstalk at the level of the tissue. We have some unpublished data on that right now that if you stimulate with like a sensory signal, we see impacts on sympathetic activity and vice versa. So we think there's definitely crosstalk at that level, but there could, of course, be crosstalk at higher levels as well. But there's no parasympathetic or vagal innervation in adipose, so especially for the vascular control, we think sympathetic and sensory are acting against each other to control vascular tone.
Great, thank you.
Darlene Sandoval, Colorado. I'm very nice presentation, Christy, as
Thank you,
always.
thank you.
Always. So I was interested in the decrease in the NANs with obesity, and I guess I have two questions related to that. One is, have you looked at a diabetes mouse model? Does it get worse once you add that layer onto that? And then the other question is, have you done the studies or have you tried to look at the literature to try to understand what the actual functional consequences to the adipose tissue when those neuronal nets reduce?
Yeah, great question. So there is a completely opposite trend with total innervation of the tissue and number of NANs. So typically when the innervation goes down, the NAN number goes up and vice versa. So it's hard to study their functions separate from the nerves in the tissue in general. But what we do think based on the data so far is that the NANs are increasing in response to inflammation. So when the tissue's inflamed, they're going up, whether it's aging, obesity. We haven't looked at other like diabetic models separately yet, but we will. And so we're testing that right now with the idea that they are nociceptors on these NANs and that this could be an inflammatory signal to the nociceptors.
Interesting. Thank you.
So unfortunately, we have to wrap up this session. I'd like to thank all the speakers for their really fascinating talks. I obviously don't think enough about the nervous system and how it regulates everything. And I'd also like to thank everybody in the audience for joining us this morning for the session.